I have been asked to talk about the newest phase 2 trials that are exciting in the management of myeloma. And what I have decided to talk is more about the difficulty of integrating new agents in the era of new molecules. Now that we have proteasome inhibitors and immunomodulators and monoclonal antibodies and histone deacetylase inhibitors, all approved. Now the patients who are eligible for clinical trials find a very difficult time in identifying drugs that are actually effective. Similarly, the regulators are finding difficulty in finding a space for the new molecules. So the first goal is to try to find a way to alter the resistance of to the unknowing agents. Particularly, there's new drugs and old drugs that have been shown that can modify the response once there is a refractory state. Interesting on that are in the IMID arena. We and others have shown that clarithromycin can change the responses and salvage patients once they have gone through a relapse state. And more importantly, when we add it to pomalidomide, the responses have doubled to the standard pomalidomide and dexamethasone. In the same vein, in this meeting and in the last ASH, nelfinavir, which is an antiretroviral drug, has also shown activity in people who have resistance to bortezomib. And although the mechanism has not been clearly defined, the responses have been outstanding. And it's suggested that it could be an overcoming multi-drug resistance, or perhaps similar to clarithromycin, it could be a pharmacokinetic effect. Being as it may, these are agents that are available and they're cheap and they're easy to integrate in any country. And in the era of cost containment, those are two drugs that are important to notice. In the newer drugs, it is promising the new immunomodulators, so I should call them the new cereblon binding agents, which are being designed and developed by Celgene. CC220 or CC122 are two different drugs that have shown potential efficacy in cell lines who are lenalidomide resistant, and the 220 has shown to be effective in cell lines and animal models who are resistant to pomalidomide. So there are those two agents that are now under development in phase 1 and phase 2 trials that have shown promise and possibly will be part of the armamentarium in the near future. In terms of resistance to bortezomib and carfilzomib, we have now also the opportunity to study the next oral proteasome inhibitor, which includes oprocimib. Oprocimib is one of the most effective drugs in vitro, and it had begun development in the clinic through different phase 1s and phase 2 trials. And there has been a hiatus in its development, but there is a new impetus to try to develop. It has been shown to have efficacy in bortezomib-resistant patients and including in carfilzomib-resistant patients, and it's an oral drug. So if we can get around the toxicity, particularly the GI toxicity, it will be a very important drug to look at in the future. Finally, the last part of my talk is going to focus on the histone deacetylase inhibitors. And although there's been a panoply of drugs with very broad effect as histone deacetylase inhibitors, the more promising, I would say, given the lack of toxicity and the efficacy, which would be the HDAC6 inhibitors, of which ricolinostat or ACY-1215 is the first one, and the next generation of that will give us the option for maybe alter the response to pomalidomide. We have seen some responses even in pomalidomide-resistant patients when you combine these agents that may not have single-agent activity, but in combination may alter their efficacy and improve response. I think it's very positive, and there is a panoply of drugs that we are looking at and that we are trying to develop. The era is a difficult time for the developers because there's now so many agents, but that's all good news for the patients. So the more we have, the better we are.